Good morning all. This is the second module of biomedical engineering. These are the contents. Electroconduction system of the heart. The heart has its own system for generating and conducting action potentials through a complex change of ionic concentration across a cell membrane. The cardiac cycle is a sequence of events that occurs in one complete beat of the heart. The two phases of the cardiac cycle are systole and diastole. Systole occurs when the heart contracts to pump blood out, and diastole occurs when the heart relaxes after contraction. During systole, the ventricles are full of blood and begin to contract. The mitral and tricuspid valves between the atria and the ventricles close, and blood is ejected through the pulmonary and aortic valves. During diastole, blood flows into the atria and through the open mitral and tricuspid valves into the ventricles. The cardiac conduction system is a group of specialized cardiac muscle cells in the walls of the heart that send signals to the heart muscle, causing it to contract. The main components of the cardiac conduction system are the SA node, sinoatrial node, the atrioventricular node or the AV node, the atrioventricular bundle or the bundle of Hess, and the Purkinje fibers. The sinoatrial node is a collection of specialized cells and is located in the upper wall of the right atrium at the junction where the superior vena cava enters. The sinoatrial node initiate the heart activity and act as a primary pacemaker of the heart. It generates electrical impulses at the normal rate of the heart, that's about 72 beats per minute at rest. This means that the sinoatrial node is said to be self-exciting. The wave of excitation created by the sinoatrial node spreads via the gap junctions across both atria from right to left at a velocity of about 1 meter per second. When both the right and left atria are completely depolarized, they contract simultaneously, which is known as the atrial systole, with blood moving from the atria into the ventricles. The rate at which the sinoatrial node generates impulses is influenced by the autonomic nervous system. After the electrical impulses spread across the atria, they converge at the atrioventricular node or the AV node located in the lower part of the wall between the two atria. The AV node acts to delay the impulses by approximately 120 milliseconds to ensure that the atria have enough time to fully eject blood into the ventricles before ventricular systole. The wave of excitation then passes from the AV node into the atrioventricular bundle known as the bundle of Hess, which is a special conduction system that carries the action potentials to the ventricles. The fibers in the bundle, known as the Purkinje fibers, after a short distance split into two branches, the right bundle branch and the left bundle branch, to initiate action potentials simultaneously in the two ventricles. This group of fibers is the only conduction pathway between the atria and the ventricles. Upon completion of impulse transmission through the Purkinje fibers, the ventricles will fully depolarize and then contract simultaneously. The sequence of electrical events during one full contraction of the heart muscle is as follows. An excitation signal or an action potential is created by the sinoatrial node. The wave of excitation spread across the atria, causing them to contract. Upon reaching the AV node, the signal is delayed. It is then conducted into the bundle of Hess down the interventricular septum. The bundle of Hess and the Purkinje fibers spread the wave impulses along the ventricles, causing them to contract. The electrical activity of the heart is recorded in the form of electrocardiogram or ECG. The electrocardiograph is an instrument which records the electrical activity of the heart. When the atria is full of blood, the SA node fires electrical signals that spread throughout the atria and cause them to depolarize. This is represented by the P wave on the ECG. Atrial systole starts when the P wave begins. The PQ segment represents the time the signal travels from the SA node to the AV node. The QRS complex marks the firing of AV node and represents the ventricular depolarization. Q wave represents the depolarization of interventricular septum. R wave is produced by the depolarization of the main mass of the ventricles. S wave represents the last phase of the ventricular depolarization. Atrial repolarization also occurs during this time. During the ST segment, the ventricles contract and pump blood. The T wave represents the ventricular repolarization immediately before the ventricular relaxation or ventricular diastole. The cycle repeats with every heartbeat. ECG provides valuable information about a wide range of cardiac disorders, such as the presence of inactive part or an enlargement of the heart muscle. The waveforms thus recorded have been standardized in terms of the amplitude and phase relationships, and any deviation from this would reflect the presence of an abnormality. These are the normal values for amplitudes and durations of important ECG parameters. Next is ECG recording system. The standard 12-lead electrocardiogram is a representation of the heart's electrical activity recorded from electrodes on the body surface. Electrical potential difference arises as the electrical impulse travels through the heart. The resulting tracing of voltage difference at any two sides due to electrical activity of the heart is called a lead. By placing electrodes on the skin, it is possible to detect this potential difference. The standard ECG has 12 leads, which are obtained by means of 10 electrodes. 6 of the leads are considered limb leads because they are placed on the arms or legs of the individual. The other 6 leads are considered precordial leads because they are placed on the torso or the precordium. There are two configurations, bipolar leads and unipolar leads. In bipolar leads, ECG is recorded by using two electrodes such that the final trace corresponds to the difference of electrical potentials existing between them. They are called standard leads and have been universally adopted. They are sometimes also referred to as Einthoven leads. There are three bipolar limb lead configurations: lead 1, lead 2 and lead 3. In standard lead 1, the electrodes are placed on the right and left arm. In lead 2, the electrodes are placed on the right arm and left leg. And in lead 3, they are placed on the left arm and the left leg. In all these lead connections, the difference of potential measured between two electrodes is always with reference to a third point on the body, which is the right leg. In defining the bipolar leads, Einthoven postulated that at any given instant of the cardiac cycle, the electrical axis of the heart can be represented as a two-dimensional vector. The ECG measured from any of the three basic limb leads is a time-variant single-dimensional component of the vector. He proposed that the electrical field of the heart could be represented diagrammatically as a triangle, with the heart ideally located at the center. This triangle is known as the Einthoven triangle. The sides of the triangle represents the lines along which the three projections of the ECG vector are measured. It was shown that The instantaneous voltage measured from any one of the three limb lead positions is approximately equal to the algebraic sum of the other two, or that the vector sum of the projections and on all the three lines is equal to 0. Next is unipolar leads. The standard leads record the difference in electrical potential between 2 points on the body produced by the heart's action. Quite often this voltage will show smaller changes than either of the potentials, and so better sensitivity can be obtained if the potential of a single electrode is recorded. Moreover, if the electrode is placed on the chest close to the heart, higher potentials can be detected than normally available at the limbs. This leads to the development of unipolar leads. In this arrangement, the electrocardiogram is recorded between a single exploratory electrode and the central terminal, which has a potential corresponding to the center of the body. In practice, the reference electrode or central terminal is obtained by a combination of several electrodes tied together at one point. Two types of unipolar leads are employed, which are limb leads and precordial leads. In unipolar limb leads, two of the limb leads are tied together and recorded with respect to the third limb. In the lead identified as AVR, the right arm is recorded with respect to a reference established by joining the left arm and the left leg electrodes. In the AVL lead, the left arm is recorded with respect to the common junction of the right arm and left leg. In the AVF lead, the left leg is recorded with respect to the two arm electrodes tied together. They are also called augmented leads or averaging leads. The resistances inserted between the electrode machine connections are known as averaging resistance. The second type of unipolar lead is a precordial lead. It employs an exploring electrode to record the potentials of the heart action on the chest at 6 different positions. These leads are designated by the capital letter V followed by a subscript numeral which represents the position of the electrode on the pericardium. The figure shows the block diagram of an electrocardiograph machine. The potentials picked up by the patient electrodes are taken to the lead selector switch. In the lead selector, the electrodes are selected 2 by 2 according to the lead program. By means of capacitive coupling, the signal is connected symmetrically to the long tail pair differential preamplifier. The preamplifier is usually a 3 or 4 stage differential amplifier having a sufficiently large negative current feedback from the end stage to the first stage, which gives a stabilizing effect. The amplifier output signal is picked up single-ended and is given to the power amplifier. The power amplifier is generally of a push-pull differential type. The base of one input transistor of this amplifier is driven by the pre-amplified unsymmetrical signal. The base of the other transistor is driven by the feedback signal resulting from the pen position and connected via frequency selective network. The output of the power amplifier is single-ended and is fed to the pen motor, which deflects the writing arm on the paper. Frequency selective network is an RC network which provides necessary damping of the pen motor and is preset by the manufacturer. The auxiliary circuits provide a 1 millivolt calibration signal and automatic blocking of the amplifier during a change in the position of the lead switch. It may include a speed control circuit for the chart drive motor. Thank you.